So we talked about media pretty much to grow bacteria. What about the viruses? What is different between the viruses and other?
Viruses need a host.
Yeah, they need a cell. That's important. They need a cell, right? Obligate intracellular parasites. So obviously, to grow a virus, you have to have a cell. And just believe me, just taking the cell out of a human or animal and just growing it, it's not such a simple task. For a long time, viruses were cultivated in animals. And it wasn't always mice. Sometimes it was hamsters, monkeys, rabbits. So you can only imagine how expensive it was to grow viruses in animals, right? In 50s, the scientist, he was Italian, of course, Renato Dulbecco, but he worked, I think he worked in England. He developed a method to grow cells. So you can take a part of tissue, I don't know, skin, fat, liver, part of tissue, disrupt it into individual cells, put them, then it was not plastic, it was glass, put them in a vessel, and the cells will stick to the surface of the vessel. Now we use plastic, so you can see those plastic flasks here, and that purple liquid is the medium. But this is the medium to maintain cells in which viruses will grow. Does that make sense? So it's kind of a complex issue. At this point, you cannot really say if the medium is complex or defined, because at this point, it's a living thing, another living thing, right? I mean, it's not a chemical thing anymore. It's a cell in which virus grows. And by itself, the medium I gave you, just, you don't have to, in this course, I don't think I asked students to memorize a single number. I don't think so. So it just shows you the medium for cell cultivation is defined. But viruses don't grow in the medium, they grow in the cell. Okay? So that's how you culture viruses. Now, live organisms as the medium. What can you use? You can use eggs. We use eggs to grow influenza. Right? And actually, eggs were used for virus cultivation before Dulbecco developed the cell culture techniques. Eggs are inconvenient. Why? What are the disadvantages? Very good. They're fragile. Okay, you have to handle them carefully. What else? They're big. Cells are big, right? Oh, no, not cells. Eggs are big. So if we talk about industrial applications, talk about thousands and thousands of eggs. You gotta build gigantic storage space. Does that make sense? What else? Huh? Well, they seed in this holder, so not really. Where do we get them from? Can you trust chickens? You cannot trust chickens, believe me. You cannot trust poultry in general. I'll give you an example. The viruses that are used in making the vaccine against influenza, every year, we get a seasonal vaccine, right? And that's the influenza virus that is inactivated. Okay, so to grow that virus, first of all, we need to put it in the eggs, and we get eggs from chicken. Well, a couple of years ago, there was a huge shortage of eggs in the United States. The reason for that was another influenza virus that infected chickens. So one influenza virus pretty much made very difficult for humans to prevent another influenza virus. Find it rather ironic. Now, these eggs that we use for virus growth, which qualities, or should I say, the biggest quality that they should have? It's a medium for the virus growth. They should be, when you inoculate a medium, they should be sterile. They should be free of the pathogens, right? You don't want your influenza virus or whatever you grow in the eggs be contaminated. Does that make sense? It's often called specific pathogen free, SPF eggs. Well, why? Now look, we understand why eggs were used in like 30s. We didn't have cell culture. Why we still use them? This answer to this question, why we still use them to grow influenza virus, answer to this question lies not in the area of science. It lies in the area of law. Why? Why do we still use eggs? To make a vaccine against influenza, for instance. Well, let's try the shoes of the pharma company. Unfortunately, we're not going to get the salary, but at least the shoes. So we are a pharmaceutical company, and we make influenza vaccine using eggs. And we decide to make the same vaccine, same virus, using cell culture. Can you just grow the virus in the cell culture, inactivate it, and put it on the market? What you have to do first? Is it expensive? Oh yeah. So if you change the medium for virus cultivation, it's considered to be a new vaccine. You have to go through all the testing. You have to get FDA approval. And the price tag, what do you think? Hundreds of millions. Hundreds of millions of dollars. Come on, nobody's going to spend $300-400 million just to get rid of eggs. Nobody cares that much. Okay, so that's why old vaccines are still made in eggs. What else can you use? You can use animals. Of course, well, not in the industrial settings, no. I can't think of anything that is grown in animals, really, any microbes. In the lab, you sometimes have to use animals to cultivate helminths, okay, to identify helminths sometimes, okay. You can use plants. That's the example of tobacco leaf infected with a tobacco mosaic virus on the right and uninfected leaf. Okay, and it's, believe me, the field of plant virology is where money is, especially in the Midwest. Infections of corn, infections of tomatoes. USDA, gives a lot of money to the labs that study the viruses or any other diseases of crops because it directly affects economy. And it's probably more than that is shelled out to people who study health-related viruses. Now, we inoculated medium, and then we incubate. The incubator, nothing really fancy. Okay. Now, why do we incubate? Try to answer this question without looking at the slide. Why do we incubate? Why do we put it at 37 degrees Celsius? What are the reasons for it? Okay, better conditions for growth. Excellent. What else? So we can cultivate microbes that microbes are in the conditions that are relevant, clinically relevant. Yes. Well, that pertains to the microbes that cause the disease. What else? They do, yeah, but what is another reason? Yep, it's controlled environment. So if you do experiment and I do experiment, if we do it at the same conditions, it's perfect. But if you say we grew microbes at the room temperature and I grow microbes at the room temperature, you can, well, at least yesterday, if you would go to the AP1 room, of course the temperature there was something like 78. And here it was 68. They're both room temperature, but they 10 degrees Fahrenheit different. Okay, and that would affect the growth of microbes, for instance. So controlled environment. That allows us to make our results to be reproducible. From time to time, we can compare different experiments at different time. And we can compare experiments done in the different labs. Does that make sense? So understand why we use incubator, why we use controlled environment, right? Isolation. Let's say you have a culture, the Petri dish, okay, with different types of colonies. And then you have to isolate each colony, right? You want to see actually if the colony is made of a single type of cells. Does that make sense? So let's say you see the little white dot. Oh, for God's sakes. Pull it out and show it to you. I'm going to grab one of those dishes and show you what we mean when we say colony. So what you do in this case, if you want to have only one of them, in this case, you have to pick this colony and isolate it. You have to confirm that it's actually a single organism. When you look at the colony, either the small or a big one, you presume that first, one cell produces one colony, and that all cells in the colony are identical. Does that make sense? That's your presumption. Okay. So you pick the colony and put it on the clean plate, the new plate. And then you streak it. Now, if you just streak it across the plate, what you may end up with is the bunch of colonies that are not really separated from one another. So you have to kind of dilute them. Does that make sense so far? Okay. So here is how you do it. You start with the streaks here shown, on the left yellow plate. Okay. And then you do it with a wire loop. There's your wire loop. Okay, so you do the streaks. Then you put the loop into the flame, you sterilize it, and then you start streaking here, diluting the bacterial population that you've got here. So you see as it gets diluted. And then you sterilize the loop again, and you dilute the population again. And then you sterilize the loop again, and you dilute the population again. So here you can see how practically indistinguishable colonies become more and more diluted to the end. Do you see that? Okay, so this is called a streak plate. We're going to do the streak plate isolation on Tuesday next week. To give you a heads up, tomorrow you're going to go around the campus and you're going to sample things from the environment. For instance, somebody, I don't know, somebody would decide to sample the nose. Okay? You put your nose sample on a TSA. Are you going to have a lot of different species? Oh, yes. And then you look at those colonies and you say, I want this one. So pick this colony and prepare a streak plate. Believe me, we're gonna figure out which colony you're gonna identify. It's gonna be your pet for the rest of the course. We don't know what the pet is, but we're gonna try and identify the microbial species. So we're gonna keep track of this. You're gonna prepare a pure culture of your pet. I'm gonna store it in the fridge, and we're gonna do different identification steps for that pet. Right? It's going to be really interesting. We identified some really nice microbes, and moreover, last semester, a couple of microbes, we never identified them. We had one, we thought it was plague, but not likely. I don't think it was plague. It looked like it, but nobody got sick. Or I don't know, they never reported to class, maybe. Anyway, streak plate allows you to thin out the initially dense population, okay, to get separate colonies. So you can see if your initial sample was actually pure. Does that make sense? Okay, can we move on? Now, other ways. You can use so-called pour plate or spread plate. Now, these methods are a little bit more elaborate, but they additionally allow you to count the number of colony-forming units, number of bacteria in your initial sample. So imagine that you have a bottle with the liquid bacterial culture. It has a ton. Well, if you want to look how the culture looks like, those two tubes. Leah, can you do me a little favor? Can you give me one? Thank you. Thanks. So this is liquid culture. See that? It's not really transparent. It's cloudy. It's cloudy because of all the bacterial cells in it. How many bacterial cells are there? I know. Can we determine it? The answer is yes. We can do that by utilizing the approach that we call spread plate or pour plate. So how we do it? Take a certain volume of that culture and dilute it a certain number of times, say 10 times. Okay? Does that make sense? Then we take 1 milliliter of that first dilution and dilute it another 10 times. And then we dilute second dilution another 10 times. So third dilution and fourth dilution and fifth dilution. So with every consequent dilution, we dilute 10 times. Does that make sense? So over the course of 1, 2, 3, 4, 5, we have a dilution factor of 100,000. Does that make sense? Does that make sense? We diluted our initial sample 100,000 times. And then from that last sample, we take 0.1 milliliter and say, put it on the agar plate. Then we use so-called hockey stick to spread that 0.1 milliliter on the surface of the plate. Does that make sense? That spreading is called, surprise, surprise, spread plate. Now, this dilution, I don't know if you can see that. I can barely see that. But trust me, there are 2 colonies on this picture. So my question to you, how many cells were in 1 milliliter of the original culture? Can we calculate it now? What do you think? Yeah, we can. Let's do it together. How about that? These 2 colonies represent 2 bacterial cells. In which volume? What was the volume that we put on the plate? The volume that we put on 0.1. So it's 2 cells in 1/10 of a milliliter. Does that make sense? 2 cells in 1/10 of a milliliter. How many cells in 1 milliliter? Yeah. Does that make sense? I always tell students, if I will tell you that there are 2 bags in 1/10 of an apple bag, if there are 2 apples in 1/10 of a bag, how many apples are in the whole bag? Just tell me 20, because apples is easy. When we move to colonies, everybody's kind of stunned. There's no difference between bacteria and apples, believe me. So we have 20 cells in 1 milliliter in the dilution 5. That's the dilution factor. So in the dilution of 100,000, there are 20 cells per ml. How many cells are in the original culture? What you have to do? You have to multiply 2 numbers. 20 by 100,000. It's going to be 2 million. Does that make sense? So, spread plate method allows you to calculate the number of bacteria in the culture, in the liquid culture. It's actually very convenient. Does that make sense? Good? I will see we may be able to try and do that. To grow some liquid culture and then calculate how many cells per mil we have there. Okay. Spread plate method, pour plate method, works similar way. The only difference is that you add your sample not on the surface of the medium, but inside of the liquefied agar. Does that make sense? You heat up the agar, it's liquid, you let it cool down just enough so it doesn't kill the bacteria. You add your sample, you pour it into the plate, it solidifies, and in a couple of days you have your colonies. Okay? So in spread plate method, you put the sample on top of the agar. In the pour plate, you add sample to the liquefied agar. Then this goes into the dish, solidifies and get your colonies. Does that make sense? Before we move on, based on the description, you have to identify strict plate, spread plate, and pour plate methods of isolation of bacteria. Good? Okay. Inspection. If you would look at this plate, I don't know how well you can see. I don't want to mess with the camera. It can be dangerous. We may lose like everything here. So if you look at this plate, what can you say about it? Describe it to me. Huh? Big circular colonies. What about, how many types of colonies? 2. The edges of the, well, you kind of cannot see from, so small colonies have clear edges, large colonies have uneven edges. If you would look at the surface, you can stop by and take a glance. Smaller colonies have perfect convex surface, right? So there are a lot of terms that you can use to describe colonies. Unfortunately, there's only one color, but you can say this colony is white, this colony is yellow, this colony is red, whatever. Does that make sense? So you can take a sample, put it on the microscope, and look at it, and say the cells are shaped as a sphere, or the cells are shaped as the rod. You can do the staining. So there are a lot of techniques. Does that make sense? Often it doesn't let you to identify the species, but it lets you to narrow down the search. Does that make sense? And finally, identification. So we're going to talk, we're going to say a few words about different identification methods that we're going to take 5 minutes restroom break and continue. How you can identify microbes. There are many ways. We're going to talk about immunological methods first. So these methods are based on the interaction between the antigen and antibody. What are they, antigen and antibody? What is antigen? We didn't talk about immunology at all, but what?
The antigen is like what's going to disrupt the virus or the bacteria. From like, essentially, it's like going to act as your antibiotic.
No. Antigen is the part of the microbe that your immune system recognizes. Antibody is what recognizes it. Does that make sense? When you receive a vaccine, you essentially receive an antigen, and your immune system produces antibodies. Does that concept make sense? So far, we're not going any deeper. That's all you need to know. The key property of antibody and antigen interaction is that it is extremely specific. It's lock and key. Antibody that recognizes influenza virus will recognize only influenza virus. Does that make sense? It's not going to recognize measles, mumps, parainfluenza, metapneumovirus, or something else, just completely. It's going to recognize only influenza. Does that make sense? Okay? That's the whole idea of antigen-antibody interaction. So then, how you can detect what is in your sample. One way is so-called Western blot. Okay, I'm going to talk about Western blot, and then we're going to take a peek into why it is called Western. It's not because it was developed in the United States. So Western blot. You have a sample of bacteria. Every cell consists of what parts? What parts of the cell do you know, chemically? Come on, let's go over it. Cells. What are they made of? Proteins. We'll stop there, because cells are made of different proteins. Different cells, different proteins. That's what makes them different. Does that make sense? So take a cell, break it in pieces, put it on the special material, should I call it? Called gel. It's actually gel. I don't know. It looks like jello. Okay. And then you apply electric current to it, and proteins of the cell gets separated. Some of them are going to be at the bottom, some of them are going to be at the top. What do you think? Which ones are going to be at the bottom? You have a group of runners. You have like really thin runners and really fat runners. And they start to run.
The bigger cells are going to be on the bottom.
Actually, no, because it's electric current that moves the proteins. So the fat proteins are going to be in the back. Oh, I didn't say current goes this way, from top to bottom. Does that make sense? So the fat, big proteins will stay up top, smaller proteins will go on the bottom. So you separate them by mass. Does that make sense? Did you ever have Jell-O? I never had, actually. I never tried Jell-O in my life. But nevertheless, Jell-O is really inconvenient thing to work with. Okay, so gel is not a convenient thing to work with. What people do next, they, using electric current, they move proteins from the gel to the membrane, special membrane. It looks like a piece of paper. It's not actually paper, but it looks like a piece of paper. Now you have proteins that are separated by mass on the piece of paper. What you do next? You add antibodies that will recognize a very specific protein to that piece of paper. And those antibodies will bind to that protein on that piece of paper. And that protein will show up as the band. The antibodies have some sort of detection. It can be like they can luminesce, or they can make a color on the piece of paper. Does that make sense? So only if the protein is present, the band is going to show up. Now, this method sounds like a lot of work, doesn't it? It actually, this method requires a lot of wet work, like bench work. If you're really lucky, you can complete the whole thing in one day. And processivity is pretty low, so you can analyze maybe 14 samples on one gel. I mean, you can make a huge gel, but anyway, processivity is low, so it's not really used in the clinical applications. It's more for research applications. It is extremely sensitive, unbelievably sensitive. It can detect picomoles of a protein. Pico means 10 in the power of negative 12 of a mole. So it can detect really low concentrations. But the downside, it's a lot of work. Another way to detect the bacteria, in this case, the antigen, bacteria, virus, whatever, is to use ELISA. ELISA stands for enzyme-linked immunosorption assay. What you do, take antigen, stick it to the surface of the plastic plate, and add antibodies to it. If antibodies stick to the antigen, you can detect them. Does that make sense? That's fairly simple. There are different types of ELISA. I'm not going to ask you those different direct, indirect, sandwich ELISA. Not really interested. But the basic concept, you have interaction between the antigen and antibody. And that complex of antigen and antibody can be detected. Does that make sense? ELISA is really simple. It has incredible processivity. Let me give you an idea. So when I did ELISA in the lab, we had a plate with 96 wells. So usually do one sample in duplicate, so you do 2 wells. Pretty much we talk about 40 samples per plate. At the same time, there were plates for 384 wells. How big is the plate? About this big. 384 wells. Now there is automated ELISA with plates for 1,584 wells. I mean, human cannot work with that. It's just impossible to remember and do stuff, but machines can do that. So processivity is extremely high for detection, and it's pretty sensitive too. Okay, so ELISA in Western blot, they use antigen-antibody interaction to detect the antigen. Now the third method that I want to mention is called immunofluorescence. So in this case you have same antibodies that detect proteins on these cells. These antibodies can produce fluorescent light, and the thing is that fluorescent light can be of different color. So this picture shows you really nicely. Green cells are astrocytes. Red cells are neurons, and blue dots are nuclei of the cells. You can see all three colors using the microscope at the same time. So for instance, if you have antibodies that can detect a virus, you can see a virus inside of the cell. Of antibodies that can detect a bacteria, you can see bacteria, I don't know, crawling on the surface of the cell. It's not quantitative, really. Okay, does that make sense? But it allows you to sometimes visualize what happens actually. Often it can allow you to visualize step-by-step processes in the cells. Before we move on, what I'm going to ask is basic principle, like Western blot, detection of a protein after separation of proteins in the gel. ELISA, detection of the antigen on the plate. Immunofluorescence, antibodies, fluorescent antibodies that detect pathogens in the cells. Does that make sense? Can we go on? Biochemical methods. If I will take a plate, pour some water in it, and add a little bit of a dye. Oh, even better, if you take some white flour, that's actually a pretty neat experiment. You take a white flour, you put it in the water, okay, live flour, okay, put it in the water, and add some green dye to the water. What's going to happen to the flour? It's going to turn green. Why? Capillary forces will pull water up the stem, you know, and the dye will reach the what's the name? Petals? Okay, petals. Chromatography is pretty much based on the same principle. You have some kind of a medium called stationary phase, and you put your sample, which consists of many different things, proteins, lipids, whatever you want to detect, on that medium. You can run your sample from top to bottom. You can sometimes run your sample from bottom to top. Okay? And then your sample starts to move through the medium. Does that make sense? Some chemicals in the sample will go faster than others. Does that make sense? Just from the general idea. You separate different chemicals in your sample based on how fast they go through the column. Does that make okay? And then you have some kind of a detector here, and you can see what your sample is composed of, or you can collect whatever you want. Right? mass spectrometry is another fascinating method to analyze things. By the way, so far, you can see that in every method, we really need to destroy the bacteria. We need to break it in pieces, right? Like here, you lyse the bacterial cell and then run it through the column. Same goes for Western blot or ELISA. Now, what is mass spectrometry? You have a protein and you break it in pieces, okay? And different pieces of the protein have different mass. Do you agree with me? Different pieces of the, you take a glass, you throw it on the floor, it shatters. Different, they're gonna be different. Pieces of the glass are gonna be different. Okay? Same goes with the protein. You shatter it, different pieces, they have different mass. If you kind of shoot them like from the cannon, which ones are gonna fly farther? Lighter or heavier? The heavier? Well, you have a dog and a hedgehog, and you kick the dog, and then you kick the hedgehog. Which one will fly farther? The hedgehog, because it's smaller, right? I didn't want to say 12-year-old and 5-year-old. It's not humane. But anyway, so smaller will fly farther. So you can separate them based on mass. Another thing that will be different between those pieces is the charge. Okay? So you put a magnet, and each of those pieces will fly differently based on its mass and the charge. Does that make sense? Because you detect both things. You kind of use both parameters to give them the unique trajectory, right? And then each piece is detected by some kind of detector at the end. And what you have in the end, you have a number of pieces, and each piece is characterized by the unique mass to charge ratio. And it turns out, if you use the same method to break down proteins over and over again, each protein will produce its own unique kind of a signature spectrum. Does that make sense? So essentially it's like breaking the glass. If you manage to hit it exactly at the same spot with exactly the same force every time with exactly the same temperature, it's going to be exactly the same glass. Every time it's going to disassemble into the same pieces. Does that make sense? It may all sound very far from clinical application, but it turns out what you can do, there are machines now. You have a patient sample and you culture. You make a pure culture of bacteria from, I don't know, urinary tract. Okay? So what you do, you take the little bit of that culture, through few additional steps, put it into the mass spectrometer and run it, and you get a unique signature of your bacteria. And you can compare that unique signature to the ones that are in the database, and it gives you immediate answer. So if the signature is 95% identical to the signature of E. coli, it's E. coli. Does that make sense? Method is wonderful. It takes 2 hours to get the signature. In the clinical settings, the machines for clinical diagnostics, it takes about 2 hours from loading the sample to producing the result. You know what's the downside? Such mass spectrometer costs about $750,000. So not every hospital can afford that. Most hospitals actually cannot. So it's the clinical labs that use it usually serve a large group of individual hospitals. I don't know if Cleveland Clinic or UH have one. They might. For instance, with Stanford University Medical Center, which serves several hospitals around, in San Francisco suburbs, they have one because they collect samples from hospitals like 30 miles away. Does that make sense? Genetic methods. Two things that we're going to talk about are polymerase chain reaction and sequencing. So what's the PCR? DNA consists of two strands, right? What is the basic major feature of those two strands? They are complementary, right? So you can make a copy of each strand, right? PCR is based on the principle of complementarity. You separate the strands, and then you add a special molecule, a special sequence of nucleotides, very short molecule that is called a primer. And this primer binds to the unique sequence in the DNA. Does that make sense? It targeted. It is targeted to the unique sequence in the DNA. Does that make sense? Think about this. It's just, I'll give you a second. Does that make sense? So say if you have a primer that is which sequence it is going to bind to? Come on. What's going to be the sequence? It's going to bind to what sequence? Based on the complementarity principle. A. There you go. Does that make sense? So it's going to bind to this sequence. Not to some other, just to this one. Does that make sense? Then you add the enzyme that starts replication of the DNA. It starts to add nucleotides to the end of the primer. Let's say so it's DNA, this is primer, okay? So when you add, and that was the primer, okay? When you add the enzyme that adds nucleotides to the end of the primer, which nucleotide is going to add? Come on, to the T end right here. Which, G, A, T, G. Does that make sense? So you get this new strand being synthesized. And then you have another primer, the second primer, on the opposite strand. So essentially I have two primers. Look at my hands. I have two primers that flank, okay, border a certain part of the DNA molecule. Does that make sense? You can select primers that recognize absolutely unique sequences that are present in, let's say, DNA of E. coli, but not in the DNA of Serratia marcescens. So they will produce they will copy that fragment that is between them. Does that make sense? They will copy that fragment that's between them. Only if you have E. coli DNA in your sample. If you do not, you can have nothing. Does that make sense? PCR is extremely sensitive. Some techniques, allegedly, can detect a single DNA molecule in the sample. some PCR techniques. It can be quantitative, okay, so it's really a nice method. The only problem with all those methods, with Western blot, ELISA, PCR, can you tell me, what is the problem? Well, more of a work intensive. PCR can be done in about 2.5 hours. Let's say I have patient's urine and I take primers that detect E. coli and I test it, there is no E. coli. Patient is still sick. Did I get any closer to the diagnosis? You look for something specific. You cannot look at everything. Does that make sense? You can find only what you look for. Does that make sense? It is. Can we do it? Can we not target? Yes. Well, there is so-called multiplex PCR. You can try and detect several things at once, but still, instead of targeting one, you target 5. Can you look at everything? The answer is yes. It is called sequencing. Sequencing is the method that allows you to determine the sequence of nucleotides in the DNA. I told you yesterday that according to Arthur C. Clarke, every advanced technology is indistinguishable from magic. So for me personally, sequencing is magic. I started to read it. I like understand words like and, or, many. That's it. I don't really get how it works. I'm not embarrassed to say. I mean, from the first read, maybe, if I'll spend a few days, I will have a gist. It's a complex method. The idea is that you have a sample, you isolate DNA, you load into the machine, and then the machine gives you all DNA sequences that it finds in the sample. All of them. Really, like all. If you have a sample with, sample of urine, it can give you sequences for 25 different species of bacteria that it finds in the urine. Awesome? Absolutely. You know what's the problem? You have a person who's sick, say urinary tract infection. For some reason, instead of treating patient, you decide to run the sequencing on his or her urine sample. And you get, 15 species of bacteria in the sample. So what? So what you going to treat? We have 15 different species. Which one is it going to treat? Yeah, that's a problem. You get, instead of not getting enough information, you get too much information. Does that make sense? So it's kind of, we don't have a magic answering machine. We don't have anything that we can put the sample in and it tells us that's the causative agent, go and treat. We still have the man, we have the woman, we have a person that interprets the results, and that's very important. That's why I tell you about those restrictions that each method has. Does that make sense? Okay. So as I mentioned, for each method that we discussed, you have to understand what it does, what it detects, what it is based on, okay? Like sequencing is determining the sequence of nucleotides. PCR is copying the very specific part of the DNA and detecting the very specific part of the DNA. Does that make sense? Am I clear on my expectations? I really want to be clear on what I expect from you. Right? Let's take a break, about 5 minutes.